Thank you so much. Yeah, this was actually a really great segue because what I'm gonna show you is some results of our long read sequencing. And basically, this is the punchline: is that using long reads and short reads together, we get better mags than the reference genomes that are in the database. Right? And so just give you maybe a little bit of context. And actually, I'll just say this was Anna Kushko's project. So this was a postdoc in my group. She had this idea. She wrote a fellowship application for it, got it funded, and somehow I just get to present it. But so we went around and we got, there's a little, yeah. So I think seems to be a little bit cut there, but we got this Shanghai pet dog sample. So these are actual pets that live with people. So you get to get their houses, collect a fecal sample, and then we had 20 GB of long reads. So this was Oxford Nanopore R10, and then 20 GB as well of Illumina data. And the pipeline was this one. So we started with, we actually had, we did a long read assembly with metaflia, and then short read polishing of the assembled genomes. And then we clustered them, or sorry, we binned them with semi-bin2, and actually using co-binning, and using this very fast stroboline-based abundance estimates that we can do nowadays. So, so in the end, we get these 2,600 mags that they replicated in 300 species. And just so to show you, here's and many of them are really just one contig mags. So some of them even it's a circular contig, but even when it's not circular, so we have a lot of these single contig mags. So that's about 40% of the data set. And then to check whether we have captured the the microbiome, we mapped reads back to our catalog, and we mapped reads from our own data set, which obviously that's the Shanghai there with the star. So we mapped basically almost everything. But then we mapped also reads from other pet dog, including a data set from Berlin that was from our collaborators, and then a couple of data sets from South Africa and the US. So slightly lower, but still we're really capturing a lot of the pet dog microbiome data set. And there we also in the plot, we see that we included other studies that have pets that live in colonies or live in shelters, and they have much lower mapping rates. And then actually in the manuscript that we're writing up, we actually go a little bit more into the that there's these different lifestyles really change the microbiome of the dogs. So today I'm focusing more on the technical side of we can get these very, very high quality mags. So I won't go into that, but it's actually quite interesting. So when we started showing this type of data, so it's great we get these high quality mags, and question that people would ask immediately is, okay, but you have long reads, you have short reads, do the short reads help? And the answer is yes, the short reads do help. So the polishing step, and show you at the top, basically it's a summary plot, and then the scatter plot has everything. But so what you see as we polish, so from fly, which is just the sort of what comes out of the assembler, and then as we polish, including first of all this sort of self-polishing step where you use the long reads to polish the assembly you got from the long reads, and then and polka being the short read, you can see that we get slightly more mags go from medium to high quality, and you have a bigger effect in the low coverage genomes. But here you can see in this scatter plot, even very high coverage genomes, there's still a little bit of an improvement. And so here we're showing the completeness difference. This is how much more complete are the metagenomes that we have. And here completeness is estimated with CheckM2. And the quality estimates are all from CheckM2. Right. And so this was great. But one of the things, and I think we saw it in a couple of talks yesterday, including Christopher Quince's talk, is this notion that when we have a short read mag, we say, oh, we can get these very high quality estimates from tools like CheckM2. And it's great when it's like 95% complete. But alas, the 5% that we miss are not the random 5% of the genome. Often the 5% that we miss are actually the really things that are really, really interesting, right? And so then when we compared our MAGs to the equivalent species in GenBank or in RefSeq, we looked at these sort of interesting things that are normally missed. So the 16S genes. So in our MAGs, we get the 16S genes much more frequently than these isolate genomes. The number of tRNAs, these mobilome sort of genes that are normally missed, and we also tend to be much more contiguous, right? And the other thing, and this was done by Alexei Chklovsky, who you might recognize the name as the first author of the CheckM2 paper. So he's now been working on how can we extract, mobile or extra chromosomal elements. So these are these circular elements that are not chromosomes. We can recover, a few hundred actually. So here we're just showing you a couple where we also get these very, very clear antibiotic resistant genes in these elements. So here's sort of as an example, there's this 13 kb element. We can recover this, and then we can also detect it not only in our study, but we can detect them in samples from across the world. Right? So we've been trying to make this data publicly available. It will soon be available. Actually, we had this internal deadline for us to have it out by this conference. We're sort of going to miss that by a little bit, but it will soon be publicly available. If in the meanwhile, if you want to get in touch, we're happy to share. We're really just finishing up the preprint soon, right? So to summarize, so we've been working on this dog data. So using long read assembly with short read polishing, we get these MAGs that are basically better than the corresponding reference genome that comes from an isolate collection. And that's because the reference genome is from short reads originally, right? So what I think is going to happen is over the next few years, all of our reference genomes are going to get replaced, right? We have very good reference genomes for a handful of species, but then all of these other hundreds of less well-studied species, we are going to get these much better genomes that are also covering, the 16S genes, are going to cover the antimicrobial resistance genes. All of this hard to assemble stuff, we are finally going to get it in the next, there's going to be this whole shift, right? We also see some relationships between the microbiome and the dog lifestyle. Happy to talk about it, even though it wasn't the focus of this talk. So as I said, most of this is Anna Kushko's work. A couple of my other students were working on this. Alex has this tool for these extra chromosomal elements. We had collaborators in Berlin. NSFC in China funded the work, and the Australian Research Council funds me. So I want to thank them, and I want to thank you.
Yeah, thank you very much. Other questions?
Yeah, thank you very much. I mean, all very intuitive results and also methods. The one that strikes out here is the high quality to medium quality after the polishing. Yeah. Can you comment on that? Because that's interesting.
So first of all, so how do we get? So we're not adding this. We're not adding any sequence, right? When we polish. So how do we get? How are the genomes more complete, right? So that's because what's happening is as we polish, CheckM2 actually doesn't check whether your genome is polished. It checks whether your gene set is complete, right? So if you have the right genes. So as we polish, we start to fix these small indels, and so we're fixing. So some genes got interrupted because got this frame shift. So we're starting to fix So we fix the genes. So this is what's happening. So you have a sequence, and because of a couple of small indels, the genes are messed up, and then now you polish, and now you get the right gene set.
Okay, that's medium quality to high quality. But what about the other way around? I mean, it's only.
Oh, okay. Sorry. I mean, sometimes the polishing does also introduce errors. So here, you can see there in the scatter plot, there are some dots below 0, meaning that the delta is negative, right? It's not a completely free lunch. There's a lot more dots above 0, but there's a few dots where the polishing, or the other thing, and Alex is also a co-author, but CheckM2 is also not perfect. So some of this could also be that CheckM2 is making mistakes, especially in the lower quality regime. CheckM2 has a lot, the error on CheckM2, so that might also be that, if you're very close to zero and in the left side, so.
One quick question and quick answer, please.
Okay, I try to be quick. Really nice talk, Luis. Very strong suggestions that we are replacing RefSeq genomes with MACs. So what you showed here today was all very convincing with the 16S copy number and so on, but one of the main problems with MACs was classically always chimeric MACs, right? So contamination. So do you think there's a complete shift in terms of quality from the long reads when it comes to contamination?
Okay, short two answers. So answer number one, I mean, some of the replacement can be by long read assembled isolates, but the second answer is there's also chimeras in RefSeq, right? And we have, it's a lower rate, but they're also there. All right, so with that.
Okay, thanks again.